Now today we will study about restriction enzymes. So last class we studied various tools needed for rDNA technology. So one by one we are going to study in detail. The first one is restriction enzymes. So what are restriction enzymes? So these are enzymes coming under a group of enzymes called as nucleases. There are two types of restriction enzymes named as restriction endonucleases and restriction exonucleases. Exo means outside, endo means inside. So what are restriction exonucleases? These are enzymes which cut the DNA from their sites. Whereas restriction endonucleases are those nuclease enzymes that can cut DNA from within the strand. So in biotechnology, they make use of more restriction endonucleases because we have to cut within the DNA strand. Okay? Now, Stewart Lin and Werner Arber in the year 1963, they isolated two enzymes from E. coli bacteria. So when E. coli bacteria was infected with or was attacked by bacteriophages, E. coli bacteria produced two types of enzymes. One was adding methyl group to the DNA of bacteriophage, thereby stopping its growth in its body. Second one was cutting the DNA of bacteriophage into fragments. What are bacteriophages? Viruses that infect bacteria. So thereby this E. coli bacteria, thereby producing two enzymes, it was restricting the growth of bacteriophage in its body. Okay, then three other scientists, that is H.O. Smith, K.W. Wilcox and T.J. Kelly, in the year 1968, they isolated the first restriction enzyme from the bacteria called as Haemophilus influenzae bacteria, and they named it as HIM-D2. So it was named as HIM-D2. So the first restriction enzyme isolated was HIM-D2. Now, they observed that this restriction enzyme always recognizes a particular sequence in the DNA and always cut a particular region, make a cut in a particular region of the DNA strand. And this particular region consisted of 6 nitrogen base pairs. ഒരു 6 nitrogen base pair ഉള്ള ഒരു particular region-നെയാണ് എപ്പോഴും ഇത് കട്ട് ചെയ്യുന്നത്. ആ ഒരു particular sequence-ലാണ് അത് എപ്പോഴും കട്ട് ചെയ്യുന്നത്. And that sequence was named as a recognition sequence. About restriction enzymes always. So each restriction enzyme has its specific recognition sequence. That is a sequence of 6 base pairs where it makes a cut. Okay? So restriction enzyme പറയുമ്പോൾ ഇത്രയും കാര്യങ്ങളാണ് നമ്മൾ മെയിൻ ആയിട്ട് ഓർക്കേണ്ടത്. And in this chapter we will be studying more about the restriction enzyme called as EcoR1. ഇതിനെ കുറിച്ചാണ് നമ്മൾ ഈ ചാപ്റ്ററിൽ മൊത്തം വരുന്ന ഒരു restriction enzymeന്റെ പേരാണ് EcoR1. So this restriction enzyme is having a recognition sequence, that is GAATTC. ഏത് ഉറക്കത്തിൽ വിളിച്ച് ചോദിച്ചാലും പറയാൻ പറ്റും. GAATTC ആണ് recognition sequence of EcoR1. What is EcoR1? It is the restriction enzyme which can be used or which is there in the E. coli bacteria. So this much is it gives an idea what is a restriction enzyme.